Let's continue talking about nutrient digestion and metabolism. Let us focus now on nutrient digestibility. Nutrient digestibility or digestibility is the portion of a nutrient that enzymes of the gastrointestinal tract are able to break down, and those nutrients are absorbed to use for metabolic purposes. How is this digestibility measured? Well, what we do is we quantify the amount of nutrients that are consumed by the pig, and that is basically we analyze the nutrient composition of the diet, and we determine how much of that diet a pig consumed, and that's how we know the amount of nutrients that were consumed. And then we measure the amount of nutrients at the end of the digestive process, and we'll have some differences that we'll see in a little bit. And then we subtract the amount of nutrients at the end of the digestive process minus the amount of nutrients at the beginning, and that's how we determine digestibility. And we'll see that in more detail in the next slide. Where do we measure this nutrient excretion, or this end? Where is the end of this digestive process? It varies for different nutrients. If we're talking about, for example, amino acids or lipids or carbohydrates, then most likely we're talking about at the end of the ileum is when we consider that digestive process to have ended. If we're talking about minerals, then we can talk about the process ends in the feces. Why do we have different sites? Because of microbial fermentation. In the case of amino acids, the microorganisms in the cecum and the column have the ability to transform lysine into arginine, and arginine into methionine, and methionine into asparagine, and asparagine into glycine, and therefore the amino acid profile that we will obtain in feces cannot be associated with the profile of amino acids that was given in the diet, whereas we can do that at the level of the ileum, where microbial fermentation is still relatively small. Minerals, on the other hand, don't get fermented. Yes, they can be transformed from one organic form to another organic form, but at the end of the day, because the way that minerals are analyzed is that we basically transform all organic forms of minerals into inorganic form of a mineral, and then that's how we analyze it. And that's why we really don't worry about any microbial fermentation, and we say that in general terms, minerals are not fermented. In order to calculate digestibility coefficients, we're gonna use here a numeric example. We're gonna say that we have a pig that ate X amount of feed, they contain X amount of lysine, and therefore the amount of lysine that this pig consumed, we're gonna say it was 20 grams. We will collect the digesta at the end of the ileum, and then we analyze the composition of that digesta, and we determine that 3 grams of lysine were remaining at the end of the ileum. So to calculate the apparent ileal digestibility coefficient, it's the 20 grams of intake minus the 3 grams that was the leftover divided by the 20 grams of intake times 100 gives us a digestibility coefficient of 85%. We call this type of digestibility coefficient apparent because they're not corrected by endogenous nitrogen losses, and we'll see that in more detail in the next slide. We do work, in order to formulate feeds, we do work with standardized ileal digestible amino acids, and we don't work, and we should not work with apparent ileal digestibility coefficients. So the difference here is that we are going to take the apparent ileal digestibility coefficients, and we're going to correct it by the amount of endogenous nitrogen losses. And furthermore, we call those the basal endogenous nitrogen losses, which are mainly used, are determined using a nitrogen-free diet that is fed to animals. The technique is basically we use different digestibility markers like titanium dioxide, like chromic oxide, and others. And in pigs, pretty much the technique that has been standardized is the T cannula at the end of the ileum for pigs. We have previously mentioned that the digestive tract will secrete a good amount of enzymes into the lumen in order to carry on the digestive process. And we have said that most of those, if not all of those enzymes, are actually proteins. And if they are proteins, they do contain amino acids. And as we see here in this diagram, then the flow of amino acids that we have along the small intestine, it's a combination of amino acids that come from the diet as well as amino acids that come from within the pig, or what we call endogenous losses. At the end of the small intestine, the microbial fermentation, it does occur and it is present, but it's present in very small amounts. Then what we have is a portion of the amino acids that come from the diet are going to be digested and absorbed, and another portion is going to be transformed into another variety of different compounds. Some of them amino acids, some of them could be just nitrogen compounds, not necessarily amino acids. The same is true for some of the endogenous losses, like the enzymes like cells, the slough off, the mucin that covers the intestine. Some of those components are going to be digested and some of those amino acids are going to be absorbed. So then what we do is, by feeding or by eliminating the amount of amino acids that come from the diet, and we do that by feeding a nitrogen-free diet, then if we eliminate the amount of amino acids coming from the diet, any amino acid that we collect at the end of the ileum of a pig consuming a nitrogen-free diet will be of endogenous origin, and we call those the basal endogenous losses. And these losses of nitrogen or amino acids could be bacteria, could be the mucin that covers the intestine, could be the digestive enzymes that we talk about. We also have the shedding and the slough of cells along the gastrointestinal tract, and another variety of losses that basically contribute nitrogen and amino acids of endogenous origin. When we talk about endogenous amino acid losses, we have basal losses, we have specific losses, and we have non-digested dietary amino acids. As we can see in this diagram here, as we increase the amount of dietary amino acid in the diet, we're going to increase specific endogenous losses and the non-digested dietary amino acids. However, the basal endogenous amino acid losses remain constant regardless the amount of dietary amino acids that are consumed. Furthermore, when we extrapolate that to a dietary intake of 0 amino acids, the amount of amino acids that we found at the end of the ileum is what we call the endogenous basal amino acid losses. At this point, it's worth to clarify, a few concepts that could be easily misunderstood or confused. One is bioavailability, and bioavailability was defined by Dr. Siebold as a nutrient is bioavailable if, on entering a living tissue, it can be used for normal metabolic functions. An example that we can use here is glucose. Glucose is absorbed as glucose, and that glucose actually can go into a cell, and that glucose can go directly into the Krebs cycle to produce ATP, for example, and so that glucose will be considered bioavailable. We talk with digestibility, but we're really concerned about the amount or the proportion of nutrients that are absorbed, because the absorbed nutrients are the nutrients that are going to reach the peripheral tissues where they're needed for different metabolic purposes. Absorption is a concept that is very easy to define. It's the amount of nutrients that go from the lumen into the portal vein. We can define that very easily. But it's very laborious and it's very hard to measure experimentally. And that's why we rely on digestibility. And digestibility is not measuring absorption directly, but is sort of indirectly saying if I place 20 grams of lysine at the beginning in the mouth of a pig and I end up only with 3 grams at the end, then I assume that those 17 grams were digested and were absorbed. And this is a big assumption. And the assumption that we do when we talk about digestibility is that we assume that the nutrients that are digested are also absorbed. And furthermore, they are absorbed in a form that is bioavailable which means in a form that can be used for metabolic purposes. Let's try to see in a graphic form the difference between digestibility, absorption, and bioavailability. Here we have the diagram of the digestive system of a pig like we had before, and we're going to continue with the same example, saying if this pig consumes 20 grams of lysine, and then 3 grams of lysine are remaining at the end of the digestive tract. So that's the numbers, or that's the information that we will need in order to calculate digestibility. So what if we want to measure absorption? How do we go about? Well, in order to measure absorption, we have to place a series of cannulas and catheters and measure blood flow of the anterior mesenteric vein and measure the concentration of amino acids that are going in that arterial vein. So I need the flow and the quantity to know how many amino acids are going into the gut. Then I need to take a sample or 2 samples of the portal vein, one at the end of the portal vein, where I'm going to infuse a marker that is going to allow me to determine the flow rate of the portal vein. And then I need to take a sample at the end of the portal vein right before the entrance into the liver, so I can determine what is the concentration of amino acids and make a mass balance and determine really what was the absorption rate for each one of the nutrients that I want to measure. As you can appreciate, this is a lot more labor intensive, and it also requires a more surgical modification in pigs than a simple digestibility coefficient. And when we talk about bioavailability, we're really talking about that nutrient moving from the lumen of the intestine through the intestinal wall of the small intestine, going through the liver, and then being able to appear in systemic circulation, where that eventually is going to go to different tissues, let's say muscle, and then that nutrient can be picked up for muscle, for protein synthesis and protein deposition, and muscle growth and the growth of an animal. Let's talk about the bioavailability of trace minerals and therefore vitamins. When we talk about mineral nutrition, most of the time we're going to talk about the availability or the nutrients or the bioavailability of nutrients in relative terms. Why do we talk about relative terms, mainly for trace minerals and even some vitamins, and we don't talk in terms of digestibility? Well, even though they are the process of digestion, absorption will be fairly comparable, the problem is that the current research methods that we have, they're not sensible enough in order to determine and measure digestibility of the trace minerals and the vitamins. We can do it with some of the macronutrients like calcium and phosphorus, but that's about it. Because calcium and phosphorus are present in larger amounts compared to the other ones, and then it allows us to be able to quantify calcium and phosphorus in the digesta in a fairly precise manner, given the current analytical methods that we have. Unfortunately, for the trace minerals and for most of the vitamins, this is yet not technically feasible. So what we do is we rely on surrogate analysis, not direct analysis. And the surrogate analysis are, for example, the activity of an enzyme that uses a specific vitamin or a trace mineral as a cofactor, or we use, for example, the amount of hemoglobin that will be tied up to the amount of iron that was digested and absorbed and used for biological purposes, for example. And therefore, for most mineral traces, we say that the bioavailability is relative to something. That something is a standard. That standard for most of the trace minerals has been selected arbitrarily as a sulfate source. So the sulfates, like a copper sulfate or an iron sulfate, those are considered to be 100% bioavailable. And any other source of iron other than iron sulfate will be compared to that sulfate and will give us a proportional rate or a relative rate of bioavailability, and we'll see that in more details in the next 2 slides. The standards for vitamins are variable, but we can follow the same principles and determine relative bioavailability of vitamins respect to a standard that is selected. Anytime we want to make a determination of or a response, whether that response is growth performance or the activity of an enzyme or the presence of hemoglobin or something else, it's important to remember that the response, as nutrient intake increases, the response is sigmoidal, or resembles an S-shaped curve. Now, for this, if we look at the proportion of the of what we will consider nutrient requirement here, if we're working somewhere between the 25th and the 75% of that nutrient intake, we can see that that line is almost linear. And that's the part of the curve that we talk about as the linear portion of the curve response. And it's the one that we ought to be working on to make relative bioavailability determinations. In this example, we're going to determine the relative bioavailability of a new zinc compound, and we're going to use zinc sulfate as a standard. So what we do is we feed different levels of the zinc compounds, both at the same level in the diets. The only difference is the source. And then basically what we should obtain is a linear response, as is graphed here. We proceed to calculate the linear regression, and we're going to do this, calculate the regression for each one of the sources with a common intercept. Once we do that, then we obtain the slope for each one of the sources. And in order to obtain the relative bioavailability, what we are going to do is we're going to take the slope of the test source, and we're going to divide that by the slope of the standard, multiply by 100, and this will give us the relative bioavailability. In this case, we determine that the slope for the test is 0.00825, and the standard is 0.105, and we do the math and we obtain a value of 78.6, and then we can conclude that the relative bioavailability of this new source of zinc is 78.6% relative to zinc sulfate. If we were to run this study again with zinc chloride or with zinc oxide, the value of relative bioavailability that we will obtain for this hypothetical new source of zinc will be considerably different. Therefore, when we talk about relative bioavailability, it is always important to know not only the number, but also what was the compound that was used as a reference standard. Let's summarize what we have discussed so far. Where is the amino acid digestibility measure, or where do we take the samples to measure amino acid digestibility? We obtain digested samples at the end of the ileum, which is the end of the small intestine. Where is mineral digestibility measured? We can measure mineral digestibility in fecal samples because, contrary to amino acids and even carbohydrates, minerals will not be fermented, and therefore we're not worried about all that great microbial processes that are taking place or will take place in the cecum and the column of pigs. When we talk about trace minerals, is bioavailability relative to what? To whatever source we select as a standard. Generally, for trace minerals, we use sulfates, but we can determine the relative bioavailability of any nutrient compared to any other nutrient that we decide to use as a standard.